Hello, my name is Shaharya Alam Chaudhary, and I'm a junior at Georgia State University, and I major in biology. Today, I'm going to introduce a topic and unpack the contents of a research paper that fits that topic. Today's presentation explores the venom's effect on the endocrine system. Now, the reason I chose this topic was actually because of a book I bought in elementary school that was all about deadly creatures. These creatures were deadly because of the venom they produced. The book was filled with creatures such as snakes, spiders, and my favorite, a Komodo dragon. Another reason I chose this topic was because of a course I took that explored animal biology. And in that course, we had a project where we had to go and find some insects and give their taxonomy. That project piqued my interest in insects, and luckily, I found a research paper that surrounds insects. The importance of this topic is that it helps me understand what exactly venom does and the different venom's effects on other creatures. To start off, we need to understand what the endocrine system is. The endocrine system essentially consists of some glands that produce hormones that play a vital role in multiple important processes such as metabolism, reproduction, and what we're going to explore today, growth and development. Venom is a substance secreted by an organism that excretes it as a defense mechanism or for hunting. In Brazil, there have been many accidents that involve catfish secreting venom on humans. This caused a team of researchers to explore why certain inflammation occurred in humans, and what they discovered was that venom played a role in how permeable a blood vessel was to molecules. The brown recluse spider uses its venom to hunt its prey, and to explore the effects of the venom, L.J. Forrester and his team conducted a set of experiments to compare the red blood cell density when there is venom present with when there is no venom present. And they found that when venom was present, nearly half of the sample lysed, meaning that there was a large effect on red blood cells. The catfish uses its venom as a defense mechanism and elicits an inflammatory reaction, while the brown recluse uses its venom to hunt by lysing red blood cells. Both of these researches help us understand the effect of venom on certain creatures, but how can a venom affect the endocrine system of another? Today, we will see an example that may answer this question. The paper I chose to explore is titled Venom of Pteromomalus puparum Induced Endocrine Changes in the Hemolymph of its Host, Pieris Rapé. The team behind this paper is displayed on the screen, and they are from Zhejiang University, which is in Hangzhou, China. Now, the hypothesis of this paper was to see if venom alone influences endocrine changes in the host, and the reason why this team decided to explore this hypothesis is because there are many other research that explored how venom with another component affected the growth of a certain species, but never explored the individual components. Now, the subjects used in this experiment were the Pteromalus puparum and the Pieris Rapé. The Pteromalus puparum is considered an endoparasitoid wasp, and the common name for the Pieris Rapé is the cabbage white. The relationship between these two organisms is quite interesting. Naturally, the Pteromalus puparum inserts its larvae into the Pieris Rapé, where the larvae consumes its blood until it is ready to claw out and prepare for its next phase of development. The caterpillar is also infected by a virus that rewires its brain to protect the larvae, which it does until it dies. This relationship can be classified as parasitism, as the larvae benefit by feeding on the host's blood. As I mentioned before, there is more than just venom injection into the host. Most of the time, the venom is accompanied by a virus. Previous experiments explored how the combination of the virus and the venom affect the growth of the host, and here the researchers want to focus on the endocrine changes the venom induces. When we talk about the endocrine system, you can assume that there are hormones involved. In this paper, the hormones that were looked at were the different types of juvenile hormones and ecdysteroids. Juvenile hormone is a hormone that prevents metamorphosis from occurring while continuously contributing to the growth of the larva. Ecdysteroid is a steroid hormone that plays a role in molting. An enzyme that the researchers were interested in was something called juvenile hormone esterase. The juvenile hormone esterase is responsible for regulating the juvenile hormones. See, now that is the necessary background info needed, so let's get to what the methodologies of the researchers were. To begin, the scientists acquired a parasitized host and the larva of the P. rapae. They incubated the host until the wasp emerged and the larva was pupated. The wasps then mated and were ready to deposit their larvae into a P. rapae caterpillar. Venom was collected by carefully extracting it from a wasp and placed it in a phosphate buffer solution. This venom was then slowly injected into the parasitized P. rape caterpillar, and the control caterpillar was injected with PBS, which is the phosphate buffer solution. Next, the hemolymph, which is analogous to blood, is extracted in 6, 12, 24, 36, 48, and 72 hours after the venom injection and was stored at -70 degrees Celsius until use. This extraction was done so the hormones could be extracted and analyzed later on. After the hemolymph was extracted, the target hormones, juvenile hormone and ecdysteroid, needed to be extracted. This was done by using 20 microliters of the hemolymph and adding juvenile hormone 3-ethyl ester, which was diluted into methanol and partitioned against 200 microliters of hexane. The methanol contained the ecdysteroid and the juvenile hormone was in the hexane phase. The juvenile hormone was then purified and concentrated under a stream of nitrogen in which 2 microliters went under GC-MS analysis, which is gas chromatography mass spectrometry. This was done to check the purity, any quantification, and confirm the structures present in the juvenile hormone. Now, the extraction of the enzyme juvenile hormone esterase is needed. To do this, the hemolymph samples were diluted in phosphate buffer and kept on ice. A portion of the diluted hemolymph was then incubated with methanol and water. The samples were centrifuged for 10 minutes and the total protein was measured by the Bradford assay. Finally, ecdysteroid was extracted from the methanol portion by vacuum centrifuge and then dissolved in a borate buffer. The samples was incubated overnight. The radioactivity was determined by a liquid scintillation counter. If you recall, the hypothesis of the experiment was to determine if the venom from the P. puparum affects the endocrine system of P. rape. What we see on this slide is some data regarding juvenile hormone 3. As you can see that the independent variable here is the time intervals that are on the x-axis, and the y-axis is the dependent variable, which is the concentration of juvenile hormone 3. The reason that the researchers only have data for juvenile hormone 3 is because there were no traces of juvenile hormone 1 and juvenile hormone 2. The top bar graph has the black bars as the pupae that were parasitized, and the white bars signify the non-parasitized. The control here would be the non-parasitized. As you can see, over time, the juvenile hormone increased over time, but around the 72-hour mark, the parasitized pupae showed less juvenile hormone than its counterpart. In the bottom graph, we see the black bars, which stand for the venom-injected pupae, and the white bars represent the control, which is the PBS-injected pupae. You can see that the juvenile hormone increases, then decreases, and becomes stagnant, whereas the PBS-injected pupae displayed increasing amounts of GH, juvenile hormone, over time. The next two graphs measure GHE activity, which is the juvenile hormone esterase, in the hemolymph of P. rape. The x-axis is the same time intervals, and the y-axis is the concentration of the GHE enzyme. So the top graph has the black bars as the parasitized, and the white bars represented the non-parasitized. The black bars seem to decrease until the 48-hour mark, which then it increased. The white bars slowly increased, then decreased, and then increased again. The parasitized have lower concentrations of GHE until the 72-hour mark. The bottom graph has the black bars representing the venom-injected caterpillar, and the white bars were the caterpillars that were PBS injected. And we can see that overall, the black bars, which is with the venom, were lower than the PBS, which was the control group. The last two graphs has the same x-axis, and the y-axis is the 20 HE equivalents, which were determined by radioimmunoassay. The black and white bars are represented by the same things as the last two sets of graphs. In this, we can see that the parasitized caterpillar and the non-parasitized caterpillar have increasing amounts of ecdysteroid and then decreases after the 36-hour mark. This pattern can be seen with the venom and PBS microinjected caterpillar ecdysteroid concentrations. The control would be the non-parasitized and the PBS caterpillars. So what do the results say about the venom effects on the endocrine system? Well, the researchers concluded that venom alone disturbs the host endocrine physiology. If we recall the hypothesis, we can conclude that the work they've done supports the hypothesis. The higher JH levels in parasitized injected pupae are high due to the fact that the larvae needs the pupae to survive, and the high levels of juvenile hormone would ensure that the caterpillar does not metamorph into a butterfly. The venom-injected caterpillar showed similar concentrations of juvenile hormone as seen in the parasitized caterpillar, so it can be assumed that the venom itself is vital for juvenile hormone production. The JHE is lower in the parasitized and the venom, since the higher concentrations would decrease the juvenile hormone, letting the organism go through metamorphosis. We also have seen that the ecdysteroid concentration is much lower in the parasitized and venom-injected caterpillar, and the trend in both are similar. The concentration of the ecdysteroid is also very similar in both, which concludes that venom alone can induce the same reaction. Overall, venom has shown that it alone has the effects of controlling a key endocrine function, growth, and development. It seems that the experiment they had done supported their hypothesis, and now is a great time to think about what's next. I believe after this experiment, they should structurally analyze the venom for its components and find the mechanisms of how the components affect the gland that produce the target hormones. After this, the researchers can experiment with similar venoms from different species and compare and contrast the effects of the venoms on a certain control species. Since this paper focused on the venom aspect, maybe the researchers can focus on solely the virus that the wasp inserts. The research papers they reference usually lump the virus and venom together, and learning about the virus would help clearly indicate its function. Does the virus solely rewire the brain, or does it also have a role in growth and development? These questions can be answered if this parasitism relationship continues to be studied. So what have I done today? What I've done is I introduced a topic that I've been interested in, gave some background information on the topic, and explained the methodology, results, and the findings of a paper that further expands mine and your knowledge on the topic. We have learned about an interesting parasitic relationship and delved into components that make this relationship possible. We came out with a better understanding of how the key component venom affects the endocrine system of the wasp's targets. Here are my works cited for my presentation. And thanks for watching and listening. I appreciate everybody for paying attention. Thank you.